right? If they have a fitness advantage, they should take over and the population should collapse. That's certainly not the case in these chronic infections. So I want to tell you first about how we do some of our experiments. I call them social engineering experiments, where we set up the conditions where sociality is required for growth and then follow what happens. And actually, I mentioned Steve in the back row here. He was the first person to do this kind of experiment. He grew Pseudomonas on bovine serum albumin. It requires LASAR quorum sensing to activate a protease called elastase, and that enables growth. And he mixed cheaters that he made with wild type and found that they could infiltrate. So this is basically the kind of result he found. LASAR mutants emerge as social cheats and populations cooperating to grow on what we use casein by using LASAR activated extracellular proteases. And we initially copied a design that was developed by Martin Schuster and graduate students in his lab. We use a strain of Pseudomonas called PA01. It's our favorite lab strain. We can inoculate a minimal broth that has casein as the only carbon and energy source. So it needs to make this LASAR activated protease in order to grow. And you can come in every morning and transfer it. It'll stay growing in log phase for about 24 hours. So continually grow in log phase, subculture, and then screen for cheaters at 5-day intervals. Cheaters would be individual colonies that don't make protease, and it's really easy to do that screen. And here's the result that we get and the Schuster lab gets. At the start of experiment, protease negative bacteria are below detection. We can detect a couple percent in the population. But after about 2 weeks, they emerge and they reach an equilibrium with the cooperators. And these protease negatives are all LASAR mutants. So LASAR mutants emerge, they don't make protease, and they can grow together with the cooperators. So I mentioned this term before, a tragedy of the commons. This is depletion of a common resource by acting rationally on one's self-interest. Well, there was no tragedy here. You can run this experiment out for a month, and for 2 months, and the cheaters and the cooperators stay in some sort of equilibrium. We were interested in why that is. And is there a way we can get the cheaters to overtake the population and cause it to crash? It would be great if we could do that in an infection. And so we thought to increase the cost of cooperation. If you increase the cost, there's a bigger incentive to cheat. And we did this just by taking away ammonium chloride. And now Pseudomonas has to acquire its nitrogen from casein, as well as its carbon and energy. And this is the question: will cheaters cause a tragedy of the commons if cooperation is expensive? My population biologist friends will show me the math that says it should. And so here's the experiment. We're looking at, again, the percent of protease negatives over time. This is in the absence of ammonium chloride. And you can see that, again, cheaters emerge after a couple weeks, and they don't reach equilibrium with the cooperators. They quickly get to about 80 to 90% of the population, and then what we call a tragedy of the commons occurs. We can't transfer enough bacteria to a new tube to get growth. And this is These tubes, I think, are pictures after 15 transfers of a control with ammonium chloride. So it is faintly blue. And this is the population after 15 days without ammonium chloride. They make a huge amount of this pigment. This is pyocyanin. It's a pigment that's controlled by quorum sensing. They also make a huge amount of elastase, about 7 times the amount of elastase that a culture at the beginning of the experiment will make. So 10% of the cells are really paying an enormous cost to keep this going. So now I want to introduce the concept of fleecing. Fleecing is a phenomenon that is inherent to cooperators. Cooperators hindering the success of cheats at some expense to themselves. So there were examples, a few examples of fleecing in the scientific literature in bacteria, but without mechanism. So what I'm really interested in is putting molecular mechanisms on social behaviors. So this is the question: why is there an equilibrium of cooperators and cheats on casein plus ammonium? What's the molecular reason? Cheaters, as I said, should cause a tragedy of the commons, even at this lower cost of cooperation. So we had several ideas. Maybe cheats aren't pure. Maybe they evolved to provide some benefit to cooperators. This was one of Ajay Gandakar's favorite ideas. Maybe proteases are partially privatized. Maybe the cooperators hold on to a little more protease and have a little more than the cheaters would have. Or maybe cooperators please cheats. And if they do this, how do they do this? So I really can't speak to 1 and 2 here, but cooperators can please cheats. And our hypothesis was that RELR, we know, activates genes for toxins, hydrogen cyanide genes, for example. And if RELR co-activates genes for resistance to toxins, and we recently learned that it does activate a cyanide-resistant cytochrome oxidase when cells are growing on casein, then the cooperators could hurt the cheats. Right? The cooperators still make cyanide, still activate the gene for the cyanide-resistant cytochrome. The cheaters don't make cyanide, and they can't resist it as well as the cooperator. That's because LASR is required to activate RELR. So we just simply did social engineering experiments, starting with a RELR mutant. So here, the RELR mutant doesn't make cyanide. And we did 6 lineages, and there was what I call a tragic ending in 4 of the 6. The open circles here are a control of wild-type Pseudomonas. You reach this equilibrium. Here are the 4 of the 6. Cheaters emerge and rise in abundance very quickly, and we lose the ability to transfer the cultures. So I'm not going to show you all the experiments to prove that in these situations, at least, cyanide was the key molecule involved in fleecing. So the real world is complicated. We heard a little bit of that in the last talk. These are not, the real world isn't a homogeneous culture. So in our homogeneous lab cultures, LASR mutant social cheats emerge. And we know that naturally occurring LASR mutants arise during chronic CF infections. But it's much more complicated than it would appear at first glance. When we first learned of this, it was the interest in trying to develop therapeutics that target quorum sensing waned very quickly. What we've learned since is that these LASR mutants are quite common. We took a close look together with Luke Hoffman, a colleague at the University of Washington. He's a pediatrician who takes care of children with CF. He was involved in a big study of Pseudomonas isolates from young children, and he thought maybe LASR mutations would be some sort of biomarker for worsening disease. So he had sequenced LASR in almost all of the isolates that they had from that study, 2,600 isolates. And 580 of them had a substitution compared to the lab strains of LASR anyway, in LASR, non-synonymous mutation. And most of those mutants were LASR null. But the big surprise when we looked closely was they all had a functional REL-R and REL-I system. Not all of them, most of them had a REL-R and REL-I system that escaped dominance of the LASR-I system. And Bree Chen in the lab decided to study one of these that was easy to manipulate genetically. And it had a LASR deletion, but it was capable of growth on casing. And these images just show this. This is our lab strain. If we make a mutation in the lab strain, a LASR mutation, this is on a medium with casing as the only carbon and energy source. So the colony grows, the elastase is out ahead of the colony, and at the edge of where there's elastase, the casing is partially degraded, and it makes the agar medium cloudy. LASR mutant doesn't grow as well. It makes a little tiny bit of elastase, so it gets cloudy at the point of inoculum. And here is her clinical isolate, E80. It has a LASR mutation, and it grows almost as well as PA01. And it also makes a lot more of this thiocyanin pigment than our lab strain. Growth on casing is dependent on the REL-R system. So here's E80 again, and if Bree knocked out REL-R, it doesn't grow very well. She can complement that. So over here is just showing that the wild type E80 and this REL-R-I mutant grow equally well in culture. This is in standard L bean broth. But if you compete them, the wild type wins out. So here, this is a competition experiment, starting with 50-50 wild type marked with one thing or not. And so this is a control, there's no advantage. But if you compete the REL-R mutant with the wild type, the REL-R mutant loses out. We do see cheaters emerge. I'm not going to talk about this much today. And the cheaters all have a mutation that disables their ability to produce this other kind of signal called PQS. PQS mutants are equally fit to the wild type. So REL-R mutants have they're disadvantaged in terms of their fitness when growing together with the wild type. So I can speculate a little bit about what may be going on in a CF1. The idea is that wild type will come in and it has all of this circuitry. I didn't talk about QSCR, so try not to let that confuse your thinking. LASR mutants will emerge. I'm not sure why. maybe they're cheats, maybe they have some sort of growth advantage. And this will shut down everything in these mutants. And they'll increase in abundance. And as they increase, the fitness of the group will wane. And some sort of selective pressure leads to the emergence of a rewired quorum sensing system. So you end up with this. REL-R and REL-I are active, and this regulon stays active. Presumably something in this regulon is important in these chronic infections. We'd like to find out what. And Ree's work shows that a mutation in REL-R will be at a disadvantage. So we never see REL-R mutations in these chronic infections. So maybe this is a kind of better target for therapeutics. There are people that have decided that's true. We're trying to learn more about it now to understand if it's true. And so that information about evolution in the chronic infections in the natural world led us back to the lab. And Maxim Kostolev wanted to see if he could rewire our strain PA01 to use REL-R and REL-I to grow on casein. And so he started with a LAS-R mutant, inoculated casein medium, and added this signal, the C4HSL, or added a strain of Pseudomonas that could make this signal. The idea is when the bacteria start to emerge in a CF lung, they'll be surrounded by other Pseudomonas making the signal. And what he found was rather quickly something emerged that could grow well on casein, and he could transfer it daily and growth would occur. If he didn't start with added C4HSL, he never got these mutants to emerge. And this just shows, this is 3 replicate experiments, and if he looks at individuals at the beginning of the experiment, some of them are able to clear a zone around a colony. These are milk auger plates, but most of them don't do a very good job of that. But if you get isolates from these evolved cultures, many more of the bacteria are capable of producing enough protease to clear large zones. And he further characterized these protease positive isolates. And he found that, like the clinical isolates, they make a lot of C4HSL. So here we're measuring C4HSL, and this is in wild type Pseudomonas, early in growth and a little later in growth. This is a LAS-R mutant that he made. They make hardly any C4HSL, and then these are 4 evolved isolates. They make almost as much C4HSL as the wild type, and they hyperproduce thiocyanin, similar to many CF isolates actually. And they make cyanide. So here's cyanide production by wild type PA01. Here's the LAS-R mutant, and here are 3 evolved strains. And I have to peek at the next slide because I can't remember what's on. So they produce cyanide, and they can fleece REL-R cheats, but the whole story isn't cyanide. And it turns out that there's just a single mutation that's required to be able to use REL-R and REL-I to grow on casein. And that mutation is in a transcription factor called MEX-T, which regulates, among other things, antibiotic efflux And we don't know exactly how that's conferring the ability to grow on casein again. So some conclusions. In our idealized homogeneous lab cultures, LASR mutant social cheats emerge among cooperators. Naturally occurring LASR mutants arise during chronic CF lung infections, but this is much more complicated and interesting than you might have guessed. Quorum sensing, I didn't tell you about this actually. Quorum sensing co-regulation of a private good with public goods affords a strong constraint on cheats. So we've learned how to constrain cheats in the laboratory. If cooperation is too expensive, the incentive to cheat increases and a tragedy ensues. This cascade allows Pseudomonas cooperators to fleece cheats, and this fleecing involves cyanide production and resistance to cyanide. We'd like to know why LASR mutants emerge in CF infections and actually in certain other types of persistent infections. We don't really know yet. It could be social cheating. I might get an argument from the back row about that. There could be some sort of growth advantage. They might be, because they don't display all of these virulence factors, they might be able to evade our immune responses better. But, in all capitals here, they're not quorum sensing negative. So quorum sensing is retained in these chronic infections, and we'd like to know why. The pie in the sky, the idea is that if we learn enough about this kind of sociality, about communication and control of cooperation, we'll be able to actually devise ways to induce tragedies of the commons in natural environments, like in infections. I told you that we learned that LASR isn't an appropriate anti-quorum sensing target in chronic Pseudomonas infections, but RELR may be. RELR mutants don't occur in these chronic infections, and we have a plausible explanation for why not. And I'll just finish with this is actually a picture of my lab and Ajay Dandakar's lab together underneath a cherry tree at the University of Washington. And here's Ajay. Here's Maxim Kostelev. I told you about his work. Li Chen is kind of trying to hide in here. And yeah, I think I'll just stop there and take any questions.